Our next speaker is Dr. Sankar Adhya. Now, Sankar and I have known each other for a long time, and in fact he is the guy who saved me when I first arrived in Madison by sending a overcoat, and unfortunately the overcoat was 3 sizes longer. Okay, but it saved me from the winter. We are known since we are undergraduate students in Calcutta, in India, and Sankar is going to talk about the puzzle of prophase lambda stability. He has been in IH for quite a few years now, and Sankar.
It's a really a privilege for me to stand here and talk to this audience celebrating the science and life of COVID-19. I'm particularly happy because one of my colleagues back home at NIH, Marshall Milbach, a peer of COVID-19, is also participating in this meeting. I'm going to talk about the puzzle stability of Prophage lab. Well, let me say that I'm working on a model system that is a development model system that's bacteriophage lambda lifestyles. Now, thanks to we know more about bacteriophage lambda life, biology of bacteriophage lambda than any other organism in this planet, thanks large part to Mark Protestant who did all the revelations. Okay, but still there are some puzzles left in lambda which needs to be solved, and I'm going to discuss one of those puzzles. It is important that we solve those remaining puzzles in lambda because many computational biologists are partly using lambda as a model system to do mathematical work. And we must solve those puzzles because the models are using many of those parameters, making assumptions which are not like by experimental biologists. They make assumptions without the actual data and the numbers. So one of the puzzles is this puzzle of Prophage lambda stability. I don't know this what happened here. Which one I use to change this? So this is explained, this ability is explained here. Dual lifestyles of phage lambda. They infect the E. coli chromosome, injected DNA, and now it makes a decision between two lifestyles. In one lifestyle, as expected, phage DNA replicates and forms all the morphogenetic proteins, and the phage host lysis and mature more phages, sponging phages come out, called lytic mode of phage growth. In the alternative mode, the phage genes are not expressed, and the lambda DNA becomes circularized and integrated specifically in the chromosome, and it's called a prophage state, and it's called misogyny. That's the second lifestyle. However, the prophage can be induced further to go to a lytic mode to reverse the pathway by some UV-SOS induction, which keeps the lambda in pressure, which blocks protein C1, which blocks the expression of the lytic genes. Now, if you don't have induction, the lysogen is very stable, that is, its stability is enormously high. Comparing to spontaneous frequency, spontaneous depression, the lack of one, 1 in 10,000 cells is spontaneously depressed, that means the lack of pressure dissociates and the promoter becomes free and transcribed by RNA polymers. In case of lambda, the spontaneous depression frequency of the Prophage lambda is less than 10⁻⁷, which is less than mutational frequency. And that is the question is why it's so low. Now, let me to answer that question or try to address that issue is Prophage maintenance. This is the partial genetic map of the region of lambda. Most of the lytic genes are expressed on two promoters, promoter to the right called PR and promoter to the left called PL. And the protein, gene C1 encodes the lambda repressor protein, which is made from this gene, and it represses the two lytic promoters PR and PL by binding to cognate operators, operator to the right and operator to the left. What happened to this one? I don't know. Well, so it should be here. And by binding those two sites. It also happens that this C1 protein regulates itself by so-called autoregulation by manipulating the promoter called promoter for the pressure maintenance, which drives this gene. Okay, the operator to the right and operator to the left region are magnified here. Actually, as Mark Prophage has shown, the OR region has 3 sub-operators. I'm a very bad graphics person. OR1, OR2, and OR3, which are surrounded by PR, the lytic promoter on the left, and PRM, the lysogenic promoter, which makes C1 on the right and on the left. Okay. Similarly, the well-PL region also has 3 operator subunits, well1, well2, well3, and on the left side is PL, the lytic promoter. C1 binds to this lead of this, binds to this side to manipulate, and C1, in summary, C1 depresses PL and PR, the lytic promoters, and auto-regulates its own promoter called PRM, and I'll go into a little more detail in the next slide. The operator elements, the sub-elements, are similar sequences as Mark has shown, and C1 dimer binds to different affinities, but also shows cooperativity. Here are the cooperative results. This is a slide I actually lifted from Mark Prothage's book called Genetic Switch. It's cooperative binding of lambda C1 protein as a dimer in gene regulation. Now, these numbers reflect relative C1 concentration. These are the 3 operator elements who are concentrated in operator to the right. Now, the intrinsic affinities for this operator for the C1 dimer protein is nearly 2 units of pressure to have saturated the OR1 site, and you need 25 units of C1 dimer to have saturated OR2, and similarly to OR3. This is the intrinsic affinities. That means this is a stronger binding site and these are weaker. Now, when you have all the 3 sites present, you have the cooperativity. OR1 needs only 1 unit, and OR2 needs 2 units, and OR3 left alone to have intrinsic affinity. So this is cooperative binding, as Mark's group has shown in many, many papers. If you mutate the OR1 site, the 2 sites down, the OR2 and OR3 sites show cooperative binding, 5 and 5 units compared to 25. In terms of gene regulation, we shown in the bottom, when OR1s, these are the 2 PR and PRM promoters, the lytic and lysogenic, and in the absence of any C1, the PR is on, the lytic genes are expressed, and PRM is off, because I'll explain that. If you have a low level of pressure C1, the PR promoter is turned off qualitatively, and PRM is still off, but if you have an amount of pressure which is present in a normal lysogen made as a prophage from lambda prophage, then the C1 at that C1 concentration is high enough to show cooperative binding. It's still PR is off, and binding of C1 dimer to OR2 site turns the PRM promoter off. That's called positive autoregulation. However, this is a positive autoregulation zone synthesis. Beyond pressure continues to go higher and higher. At some concentration, this C1 leftover operator site OR3 is occupied and is saturated. When that happens, the PRM promoter is turned off. Okay, I should mention, I'll come back to these points. In these experiments, in Mark's, all of his papers, they are dealing with this DNA fragment and the well region, the distal promoter, which is promoter and operator segment, which are located 3 kilobase pairs away, was deleted, was absent. Now, this is Mark's model. That is, the PR lytic promoter is always on when all 3 sites are vacant. As you increase the PR C1 dimer concentration, these 2 sites are filled at a lysogenic concentration of C1 and is activated, and PRM at higher concentration when all 3 sites are occupied, PRM shut down. There's orthogonal regulation, both activation as well as suppression, but on the other hand, PR promoter, the lytic promoter is depressed when these 2 sites are occupied for a time. Now, like I mentioned, in this case, what I described as the lysogenic situation in a stable prophage state, that the OR1 and OR2 sites are occupied by 2 dimers and forming a tetramer because of cooperative binding. here protein-protein interaction, and this side is similar thing on the left side. But I mentioned that in all of the models built on, in the absence of this region, 3 kilobase pairs away. Now the question we asked is that would there be any effect in a normal wild type situation if this region is present in the prophage, as it is in normal prophage state? We did some in vivo experiments by fusing the PR to laxate reporter gene and PL to luciferase lax reporter gene on this side, and summarized, is all published. The deletion of where in this region partially depresses the PR promoter, although the pressure is present in normal amounts. And on the other, similarly, on deletion, when you delete this region, the PL promoter, as by luciferase, is also partially depressed, about 2-fold to 3-fold, not very much in these cases, okay? Suggesting that there is an influence of the distal operator, respective distal operator, in the presence of the two PL promoters. So we concluded that must be an interaction of these two guys forming a tetramer, and there is a DNA loop structure. Now, we'll go to something called 8-mark DNA loop proposal, and this similar conclusion is made by Barry Egan from Australia and Benomuller Hill from other kinds of experiments. I'm not going to detail. So that was in vivo. That is, we did this influence of one promoter operator complex by the distal model. Okay, we want to test that in vitro. We did that. Now the DNA, these are all in vitro transcription assays in purified system. We have a piece of DNA, circular plasmid DNA, where we have the PR promoter, PRM promoter, and PL promoter, the corresponding operator sites all present, except that we put by chlorine thermometer, transcription thermometer, one here, one there, in different distances from the corresponding promoter, so we have discrete size messages we can follow in the gel. And the transcription is also shown here. This is the increasing concentration of C1 dimers, and you can see without any C1 protein, you have a high level of PL and PR transcription as judged by in vitro transcription assay. This is a control promoter which are not affected, and as you can see, the increasing concentration C1, both PL and PR are repressed as was expected. Now, as was also expected, the PRM promoter, which is very low without any C1 protein, goes high and comes back at higher concentration, as was found by Mark many years ago. Now, because the wild type PRM promoter is very weak in vitro, it is hard to quantify these regions when the level is low. So we used one of the mutants of PRM up, which means the promoter has an up mutation which makes higher level of message without the C1 protein, and it also follows the same rule quantitatively, always a higher level of promoter, but you see it's also activated, repressed, just like the wild type. So in many of the experiments that we use this promoter for quantification. And the conclusion that C1 represses PL and PR and autoregulates PRM as expected, at least qualitatively. Now this is the comparison of our results in vitro assay from the previous slide and in what was modeled by Mark from in vivo results. As you can see, the two results, these are PL and PR promoter. They are repressed quickly and remains repressed tightly, and PRM goes up and comes down, which mimics this picture. Although this was done without PL being present, or whale operator being present here where was absent and where was present there. Now we wanted to test that influence by using some mutant operator. These are the sequences of the 6 operators, well 1, 2, 3, and OR 1, 2, 3, and it's shown in this way. That is, there's a diad symmetry. On the left part of these sequences have consensus sequences shown by this box, and these are essential bases, and we mutated these 2 consensus CC to AT sequence to create mutation in different situations, either mutate well 1, well 2, and so on and so on, okay, to see the effect of individual mutations. By DNA binding assay, I should say, when we make these changes, DNA binding is totally gone from each of these operators. Okay, now the effect of DNA looping on the pressure pair. The model suggests these 2 guys see each other to bring some change in some regulation. That was the test. And we mutated, for example, if we mutate all the three operators in a given area, then they should be unable to interact with each other. That was the idea, and we tested that. But if you mutate all three elements of the ward side, you can see there is hardly any effect of PR depression. Of course, PR is very depressed because there is no active operators there. There is might be marginal effect between these two. Careful quantification. Similarly, the depression of PR by C1 protein is hardly affected on your mutation well, which speaks against the DNA loop model for the time being. Okay, so we conclude that the DNA looping has marginal effects on PL and PR depression white. It has some effect, but very marginal. Now, in lysogen, the amount of C1 present always varies from cell to cell, as is shown by Stanley Brown. So it fluctuates. We want to see the state of depression when there is a lowering of C1 level for physiological reasons or stochasticity or by sudden arrival of a mutation in a given operator. What happens then? So we mutated in this template, we mutated the wall is completely mutated, the well area or well operators, and we have this ward, the site 2 is mutated, okay? So now PR depression depends entirely on site 1 bonus, okay? And the results are shown. In the absence of well operators, PR is never depressed, and PR is depressed only at high concentration because only one of the operator elements is present. But you bring back an active well region, you can see that depression is restored quite a high rate, okay? So you conclude when the first level binding is lower by either by mutation or by lowering C1 concentration, there is an effect of DNA looping as concluded here. Similarly, I'm not going to detail that if you do the same thing at the well, that is, you remain only one operator to have an effect on PR depression on the left side, and in the absence of WAR, there is no PR depression. You see very small amount of depression, but if you bring back the ability to loop, then you restore depression quite a bit, suggesting DNA looping tightens depression at PL and PR operator or promoters when the depression amount is low or the operators are mutated, partially mutated. Okay, so that is a conclusion. I'll come back to that. Effect of DNA looping on PRM autoregulation, I mentioned that. Now, I said that this PRM is activated, initial concentration is depressed, but if you eliminate, that's because of binding of C1 dimer to WART3. If you eliminate WART3 by mutation, you see normal activation because it binds to WART2, but you do not see depression. That is consistent with the potassium model. Okay, and in this case, you don't see much effect on PL and PR depression. However, if you eliminate the well operator and you have normal alleles of WAR3 is present here, you see activation, but you don't see depression. So, affecting or eliminating the distal operator size 3 kilobase pairs away does not affect positive autoregulation of C1 of its own synthesis, PRM promoter, but does affect the autorepression. Okay, so here. More interestingly, that you can mutate the individual well-3 site, which is located farther away, you do not affect positive regulation, but you eliminate the repression. So the repression of PRM by C1 by binding to WART3 sites is heavily affected by a mutation elimination of well-3 sites, which is located 3 kilobase pairs away. So we propose this following model, that is at lysogenic concentration of repression at in a normal prophage, you have repression dimers bound to each WAR1 or WAR2 site by cooperative binding from a tetramer. Similarly, the well1 or well2, this has been known before, and we proposed initially that these two tetramer interact to say an octameric loop. Okay, that brings back slightly, affects PL and PR depression marginally. But when depression concentration goes higher because of positive autoregulation, because the well-3 binding affinity, intrinsic affinity, is much higher than the others who are 3, then the pressure comes and binds here. This recruitment of C1 dimer to R3 distal site helps bring back by a cooperative binding of C1 dimer to R2, and that causes positive auto-repression. Negative autoregulation of PRM by C1. That's our model, and that is considered easy. And a similar conclusion has been reached, the effect of WEL3 in controlling autoregulation, negative autoregulation of PRM by C1-2 by Page biology, mutation experiments life cycle by Barry Hill's group. Now, there are two kinds of loops I said. That is, one is an octameric loop, another is a potentially octamer plus a tetrameric loop. Okay, and the question is their state, the stability of these two loops. So we do a single molecule stability assay of DNA loop by tethered particle motion. This is explained here. Now the DNF is carrying the two operator regions, 3 kilobase pairs apart, are tethered on a grid, and the other end has a magnetic beat which can be monitored, and this has a Brownian movement around it all the time without the protein. It should move around, the limitation is attached here, and if you locate them at varieties of times, you see these dots and it's kind of semi-sphere, and the outer diameter or radius of this region, if you tell the length of this piece of DNA. Now, if you have the pressure bound to that, if that's form a loop, then the diameter or radius will decrease from this side to a smaller side. And if you follow the length that is in an unlooped state, the length will be higher, and in a looped state, the length will be lower, and it goes on and on, up and down, giving some signal if there is a fluctuating DNA loop. So these are the sample results of TPM tethered particle motion analysis with no C1 concentration. You see this, that the bead remains at a position which is consistent with the length of the DNA that was added in the designed experiments. But if you get a high level of C1 repression concentration, then you see a length is reduced, and this difference in length is consistent with what was expected from the size of DNA present in that loop, which is 3.2 kilobase pair apart. Okay, and if you have an intermediate concentration of C1, you see the fluctuation forms a loop and comes back, unloops and loops and unloops and loops and transition level. And we did many other concentrations, and the results are summarized in the next slide, and that is here. So when it's white type situation, no C1, the results are shown here. So we measure the frequency, do the experiment many times, and you do the frequency of their loop state or unlooped state here versus the length. This is, we're following the length. And without C1, in the white type situation, you see the distribution, the frequency distribution is consistent with this being unlooped. That is what is expected. However, at low level of C1 concentration, like 20 nanomolar, you see half of the molecules at an unlooped state that happen in loop state. This is the frequency distribution. If you increase the C1 concentration from 40, 80 to 70 and so on, you shift this peak more to the loop state, as is shown here. Okay. However, if you mutate these two operator sites, WER3 and WEL3, which is the sample results are shown here, then at 20 nanomolar, 60, 40, 20, 40, and 80 nanomolar concentration, you see the DNA remains mostly at the unlooped stage, and very few goes to the loop stage, suggesting that in absent, although tetrameric loop formation is possible, it's very unstable. It doesn't remain in the loop state. That's the major conclusion. Okay, and next slide shows that the distribution loop frequency. When you allow the octameric loop, it is very stable, the frequency is very high, but you disallow by mutating the WEL3 and WER3 mutation, the loop probability is much smaller, and this loop probability is a positive delta G, that means it's not favorable. However, the loop frequency with the tetramer plus, this is reverse, sorry, and the octameric loop has a negative delta G, and the octameric loop has a positive delta G, and the dual-mired loop has a negative density, which is more stable. So last slide shows, the few slides shows our conclusion. That we said PL and PR repression, which determines the Prophage stability of the looping, originally helps PL and PR, marginally helps PL and PR. It shields PL and PR repression against lowering C1 concentration or operator mutations, etc. Multi-level cooperative DNA loop discourages C1 dissociation from the operators. But Morelli et al. Suggested that this loop is very stable from mathematical viewpoint, so we find in real life this dual-mired loop is more stable than the octa-mired loop. Second conclusion is PRM regulation. Looping does not affect PRM activation, as was suggested by Anderson from mathematical calculations. We don't find that, but it's essential PRM repression, as was suggested by biological experiment from Barry. And that is what I mentioned. And the last conclusion is that loop stability, we find octa-mired loop, WAR1, WAR2, and WAL1, WAL2 by octamer formation to C1 dimer is energetically unfavorable. Loop is stabilized by addition of WAR3, WAL3 contact. And so the last one is an experimental conclusion. So we believe that the DNA looping in lambda is not an evolutionary declaration, but an epigenetic strategy to bring Prophage stability by a tremendous amount. And most of the work I discussed was done by Del Lewis in my lab, and we did the single TPM analysis of single DNA molecule and collaboration with Laura Finzi at Emory University. Thank you.